Imagine a highly advanced survival compass.
Okay, a compass.
Right. Evolution designed this incredibly sophisticated instrument within your brain, and its sole purpose is to point you towards the things that keep you alive, Things that ensure the continuation of the species.
Ah, like caloric density, hydration, social connection.
Exactly. Reproduction, all of that.
Mm.
The needle is calibrated perfectly to your environment. But what happens when a chemical pathogen hacks that compass?
It gets into the hardware.
Yeah, it rewrites the underlying software so that the needle only points to one destination, completely and forcefully overriding every other innate survival instinct you possess.
Right, because when you look at it that way, a substance use disorder, well, it isn't a broken moral compass at all.
No, not at all. It is a hijacked biological one.
It's a profound hijacking, really, because the substance fundamentally co-opts the exact neural pathways that evolved to guarantee our survival. I mean, we are looking at a system that is operating exactly as it was designed to.
But it's responding to an artificial supernormal stimulus.
Spot on.
And that distinction changes everything about how we approach this in the clinic, doesn't it? Evolution built the compass. But today we're looking at what happens when the needle is captured. So welcome to our deep dive. We are synthesizing Chapter 1.31 of Kaplan and Sadok's comprehensive textbook of psychiatry, really dissecting the neuroscience of substance use disorders.
It's a heavy chapter.
It is, yeah. So for you listening, whether you're a psychiatry trainee, a registrar on the wards, or just a clinician seeking to refine your neurobiological models, our mission today is to tear down that artificial wall between the lab bench and the consultation room.
We want to map the journey from the molecular mechanisms, deep within the synapse, all the way to the clinical manifestations you see sitting right in front of you.
Exactly. And that journey really demands that we leave behind some outdated vernacular, right?
Oh, absolutely. The field has largely moved away from the culturally loaded term, drug addiction, and embrace the DSM-5 classification of substance use disorder, or SUD.
Which isn't just an exercise in academic semantics.
No, not at all. I mean, the term addiction is quite binary, and it's heavily fraught with stigma. Conversely, the older medical term, drug dependence, is biologically imprecise.
How so?
Well, think about it. If a patient takes prescribed opioid analgesia for an orthopedic injury for, say, 6 weeks, their physiology adapts. They develop tolerance.
Right, and if the medication is abruptly ceased, they'll have a physiologic withdrawal syndrome.
Exactly. That is dependence. It's an expected, completely normal biological adaptation. It definitely does not mean they have a psychiatric disorder.
So we're really looking at a measured continuum here. SUD evaluates this whole constellation of cognitive, behavioral, and physiological symptoms.
Right. It assesses impaired control, social impairment, risky use, alongside those pharmacological adaptations.
So when a patient meets 6 or more criteria, we are diagnosing severe SUD.
Yes, which is a relapsing central nervous system disorder. It's defined by compulsive drug seeking and a profoundly negative emotional state when the drug is removed.
Which brings us to the real anchor of this entire deep dive. Psychiatry is essentially the study of how the brain, the mind, and experience interact, right?
And how that brilliant process can go horribly wrong.
Yeah. We have to move past the what of the diagnostic criteria and relentlessly question the how.
And to understand that how, we really need to look at the epidemiology of exposure versus vulnerability.
Right, because they aren't the same thing.
No. The text highlights that roughly 15 to 16% of the adult population in the U.S. engages in illicit drug misuse at some point. However, only around 3% go on to develop a substance use disorder.
So exposure to the chemical pathogen is a prerequisite, naturally.
But vulnerability to hijack is not universal.
That 12% gap is the grand mystery, isn't it? Like, what is happening in the brain of the 3% that transitions them from casual recreational use to a severe locked-in SUD?
Well, we conceptualize that transition as a fundamental shift from impulsive behavior to compulsive behavior.
Okay, impulsive to compulsive.
Yeah, and to understand that shift, we have to examine the changing motivational drivers. In the early stages, substance use is entirely impulsive. It's driven by positive reinforcement.
So they take the drug, experience a hedonic reward, a euphoria, or a high, and that positive stimulus reinforces the action.
Exactly. They're acting in anticipation of a reward.
The honeymoon phase, essentially.
Right.
But if we talk to patients with severe, protracted SUD, they'll often tell you that the drug hasn't actually been fun for years. The euphoria is just gone.
And that brings us to the crux of the pathology. The driver has shifted from positive reinforcement to negative reinforcement.
So they aren't using the drug to achieve a high anymore.
No, they're using it compulsively to remove an aversive state. They are medicating against a crushing dysphoria. And this transition is elegantly mapped by Solomon and Corbett's opponent process theory of motivation.
Oh, let's really dig into this, because opponent process theory seems to explain the cruelest irony of the whole disorder.
It really does. The theory basically posits that the brain is relentlessly protective of homeostasis.
Right.
So when a drug introduces a massive artificial hedonic spike, which we call process A, the central nervous system immediately deploys a counteracting biological opponent process.
Process B.
Exactly, process B, to drag the system back down to baseline.
So the brain essentially throws out a physiological parachute to slow down the ascent.
That's a brilliant way to put it. Early on, process A is enormous, and process B is small and somewhat delayed.
So the high is intense and the come down is relatively mild.
Right, but neuroadaptation completely changes the math. With chronic exposure, the receptors downregulate, and the hedonic impact of process A diminishes. You develop tolerance.
But process B, that anti-reward parachute, grows massive.
It does. It deploys faster, and it lingers long after the drug has cleared the system. The baseline of the patient's emotional state is permanently dragged downward into a state of allostatic load.
Wow.
So they are no longer chasing a high, they are desperately fleeing a biologically enforced state of misery.
That maps perfectly onto the three-stage cycle of addiction outlined in the chapter.
It does, yeah.
Stage 1 is binge and intoxication, driven by that initial impulsivity and reward. Stage 2 is withdrawal and negative affect, where that massive process B really takes over.
Right, and then stage 3 is preoccupation and anticipation, which is driven by craving, cues, and compromised executive function.
So that cycle is going to be our structural spine today. But before we look at the physical neuroanatomy, the text presents 5 major theoretical models of addiction. Let's synthesize these, because they aren't competing theories, are they?
No, not at all. They are more like overlapping lenses examining the very same pathology. The foundational lens is the psychomotor stimulant theory.
Which establishes what, exactly?
That all addictive drugs, despite having wildly different chemical structures and primary mechanisms of action, they all eventually converge on a final common pathway.
Ah, the mesocorticolimbic dopamine system.
Exactly. This system is the brain's natural approach circuitry. When it's activated, it promotes approach behaviors towards whatever the stimulus is.
Okay, so that establishes the fundamental wiring. But the second model, incentive sensitization theory, this is where the cognitive distortion really becomes apparent.
Right.
Because this theory uncouples the concept of wanting a drug from liking a drug. I was trying to conceptualize this for a patient once. Imagine a giant flashing neon sign for a restaurant.
Okay, I like this.
The sign represents the incentive salience, the wanting. You see the sign, you go in, and the food is phenomenal, you like it. But as the disorder progresses, the food inside starts tasting like absolute ash.
You despise the food.
Exactly. Yet the neon sign outside keeps getting brighter, larger, and more aggressively hypnotic every single day.
Yeah.
The salience machinery is just screaming at you to go inside, completely overriding the reality that the reward is gone.
That analogy captures the tragedy of incentive sensitization perfectly, because the neural circuits that assign salience, that wanting you mentioned, they become hypersensitized with repeated drug exposure.
They become wildly reactive.
Very much so. Not just to the substance itself, but to the environmental cues associated with it, the neon signs.
Right.
And this sensitization occurs entirely independently of the hedonic liking circuits, which are simultaneously developing tolerance.
Which explains so much. This is why a patient can experience overwhelming, debilitating craving for a substance that they consciously know will bring them nothing but ruin and misery.
Exactly. Which flows directly into the third lens, aberrant learning theory. Because if that neon sign is blindingly bright, the behavior of walking into the restaurant,
it completely ceases to be a choice. It just becomes a reflex.
Spot on. Aberrant learning theory describes the transition from goal-directed, voluntary action to automatic compulsive habit. And we can actually map this neuroanatomically.
Where does it happen?
Well, initially, drug seeking is mediated by the ventral striatum, which evaluates reward and emotion. But over time, control shifts dorsally to the dorsal striatum.
Which is the region responsible for ingrained motor habits.
Exactly. And the learning is termed aberrant because the habitual behavior becomes entirely decoupled from the outcome. The patient continues the behavior despite severe negative consequences.
Like loss of employment, destruction of relationships, failing health.
Right. Normal learning dictates that punishment extinguishes a behavior. In SUD, the dorsal striatum basically renders the behavior immune to punishment.
I often hear families ask, why don't they just use their willpower? They know it's destroying their life. And that brings us to the fourth model, doesn't it? Frontostriatal dysfunction theory.
It does.
Because the willpower argument assumes the patient has a functioning set of brakes.
And that's a biologically flawed assumption, really. Chronic substance use physically alters the top-down executive control networks originating in the prefrontal cortex. We call this cortical hypofrontality.
So the basal metabolic activity in the frontal cortex is significantly reduced.
Precisely. The networks responsible for response inhibition, delayed gratification, and evaluating long-term consequences, they are fundamentally compromised.
So let me get this straight. You have a hypersensitized drive to seek the drug, an automatic motor habit executing the behavior, and a prefrontal cortex that is just too offline to hit the brakes.
A jammed accelerator and severed brake lines.
Wow. And the final theory, hedonic allostasis, paints the darkest picture of what it feels like to sit in that runaway car. We mentioned the brain's attempt to maintain stability through change, which is allostasis. But the baseline doesn't just return to normal after a binge.
Oh, no, normal drops. The hedonic set point is radically lowered, resulting in a state the literature refers to as hyperkataphia.
Hyper what? Hyperkataphia. It sounds like a curse.
It essentially is. It derives from the Greek for extreme dejection or sadness. It's a protracted, persistent state of profound emotional distress, hyperirritability, dysphoria, and just crushing anhedonia.
So the patient in this allostatic state is not pursuing a high.
Not at all. They are using substances merely to drag themselves out of an agonizing emotional deficit, literally just to feel a fleeting semblance of normal.
That is incredibly bleak. So, all right, we have the conceptual models. Let's move from the theories to the actual neurocircuitry. Let's map stage 1, binge and intoxication, which centers on the reward circuits. Where are we in the brain, and how do vastly different chemical structures pull the exact same dopaminergic levers?
Okay, so the epicenter of acute reward is the projection from the ventral tegmental area, or VTA, in the midbrain, to the nucleus accumbens, or NAC, in the ventral striatum.
Okay, VTA to NAC.
Right. But we really have to elevate our understanding of dopamine beyond the pop science idea of a pleasure juice.
It's not just the happy chemical.
Exactly. Dopamine neurons have two distinct modes of firing, tonic and phasic. Tonic firing is a slow, steady background release of dopamine. It sets the baseline motivational state and the overall tone of the system.
It dictates how much energy you have to pursue goals generally.
Correct. Phasic firing, however, involves rapid, high-frequency bursts of dopamine release, and this is the teaching signal.
The teaching signal. So phasic bursts encode the prediction error of reward.
Yes. If you experience something unexpectedly rewarding, you get a massive phasic burst, which stamps in the memory and assigns intense salience to the stimuli that preceded the reward. Addictive substances just aggressively hijack this phasic firing.
Let's look at the pharmacology of that hijack, because the mechanisms are wildly different. If we compare cocaine and heroin, they both flood the nucleus accumbens with dopamine, but they do it in completely different ways. I tend to think of cocaine as a direct, blunt force trauma to the synapse.
That's fair. Cocaine is a psychostimulant that operates directly at the terminal button in the nucleus accumbens. Normally, you see, after dopamine is released into the synaptic cleft, it's quickly vacuumed back up into the presynaptic neuron by the dopamine transporter, or DEET.
Right.
Cocaine physically binds to and blocks the DEET. The vacuum is broken.
So the dopamine simply pools and accumulates in the synapse.
Exactly. It repeatedly agonizes the postsynaptic receptors, leading to massive sustained dopaminergic signaling.
So cocaine traps the dopamine in the cleft. What about opioids, though? Because they don't touch the dopamine transporter at all.
Opioids operate further upstream, back in the ventral tegmental area. Within the VTA, you have the dopaminergic neurons, but they are tightly regulated by local GABAergic interneurons.
And GABA is inhibitory.
Right. These GABA neurons act as a continuous inhibitory clamp. Their resting potential keeps the dopamine neurons quiet. Now, opioids, like heroin or fentanyl, bind to mu-opioid receptors located on those GABAergic interneurons.
And the mu-opioid receptor is an inhibitory G-protein coupled receptor.
Exactly, it's G-coupled. So when the opioid binds, It inhibits adenyl cyclase and opens inwardly rectifying potassium channels, GRK channels.
Potassium rushes out of the cell.
Yes, deeply hyperpolarizing the GABAergic interneuron. And because the interneuron is hyperpolarized, it completely stops releasing GABA.
So you inhibit the inhibitor. You silence the silencer.
Exactly. The inhibitory clamp is removed, and the VTA dopamine neuron is suddenly free to fire rapidly, sending a massive surge of dopamine down into the nucleus accumbens.
That is the precise mechanism of opioid-induced reward. That makes perfect sense. Now, you brought up alcohol earlier. The textbook mentions that alcohol is phenomenally messy as a pharmacological agent.
Oh, it is. It doesn't have a single specific receptor like cocaine or heroin.
Right.
It acts as a positive allosteric modulator at inhibitory GABA-A receptors. It acts as an antagonist at excitatory NMDA glutamate receptors, and it even triggers the release of endogenous opioids and endocannabinoids.
It's basically just throwing biochemical spaghetti at the wall, isn't it?
It is, yeah. But the result proved the final common pathway rule perfectly. All of those diverse, messy actions eventually converge on the VTA to inhibit those same GABAergic interneurons, or they directly excite the dopamine neurons.
So the route is infinitely more convoluted, but the destination is identical. Supernormal dopamine release in the ventral striatum. All right, so the synapse has been flooded. The binge concludes. The substance metabolizes and clears. Now we enter stage 2, withdrawal and negative affect. The textbook describes this not merely as the absence of reward, but as the active recruitment of the anti-reward system.
Right, and this requires us to look at two distinct neuroadaptations, within system and between system.
Okay, let's start with within system.
The within system adaptation is the brain's defensive response to that massive dopamine flood we just discussed. To protect itself from excitotoxicity and overstimulation, the striatum aggressively downregulates the expression of dopamine D2 receptors and decreases the natural tonic release of dopamine.
The system becomes profoundly hypofunctional.
Yes, the individual is left in a hypodopaminergic state. This explains the profound anhedonia.
Right, so natural rewards like a beautiful sunset, a great meal, the embrace of a loved one, they simply fail to register.
Exactly, because the baseline dopamine tone is too low and the receptors are just depleted.
But the absence of joy is really only half the torment. The between-system adaptations are where the active suffering of hypercataphia is generated. And this involves a region called the extended amygdala.
Yes, the extended amygdala. It's a conceptual macrostructure. It encompasses the central nucleus of the amygdala, the bed nucleus of the stria terminalis, or BNST, and a transition zone in the shell of the nucleus accumbens.
And what does this circuit do?
It's the major interface between stress, fear, and reward. During substance withdrawal, the extended amygdala is bathed in stress neuropeptides. The primary culprits here are corticotropin-releasing factor, or CRF, and dynorphin.
Dynorphin. It sounds like the sinister mirror image of an endorphin.
It effectively is.
Yeah.
I mean, endorphins bind to mu-opioid receptors to produce analgesia and euphoria, right?
Right.
Well, dynorphin binds selectively to kappa-opioid receptors, or KRs. When dynorphin activates KRs, it produces profound dysphoria, aversion, and depressive-like states. It directly inhibits dopamine release.
So withdrawal isn't just an absence of dopamine. It's an active, biochemically enforced state of misery driven by a CRF and dynorphin storm within the extended amygdala.
Exactly. Which catapults the patient into the third stage, preoccupation and anticipation. The intense craving phase.
We touched on this with aberrant learning theory, transitioning from the ventral to the dorsal striatum. How does the anatomy actually pass the baton from emotion to habit?
It's accomplished through an elegant, albeit destructive, neuroanatomical feature known as spiraling loops.
Spiraling loops.
Yeah, the circuitry connecting the prefrontal cortex, the striatum, and the midbrain. Well, it isn't a series of isolated closed circuits. It's a feed-forward ascending spiral.
Okay, walk me through the spiral.
So information from the ventral striatum, the reward and emotional center, projects down to the midbrain dopamine neurons. But those midbrain neurons don't just project back to the ventral striatum. They project slightly more dorsally.
So it cascades upward.
Exactly. Activation in the limbic, ventral sector feeds forward into the cognitive sector, which feeds forward into the motor dorsal sector. Over years of chronic substance use, this ascending spiral entrenches the behavior.
Meaning the cue is perceived and the signal skips the prefrontal evaluation completely.
Yes, it spirals directly into the dorsal striatum to execute the automatic drug-seeking motor habit.
Wow. Okay, so we've mapped the circuits, but here's the grand clinical dilemma. A patient enters a detox facility, Over 10 days, the substance is metabolized, the acute physical withdrawal subsides, the physiological dependence is completely broken, they leave the facility. Why does the vulnerability persist for months, years, or even a lifetime?
That is the big question.
To answer that, we have to zoom past the systems and circuits, right past the synapse, and dive right into the nucleus of the neuron. We have to examine intracellular signaling, structural plasticity, and epigenetics.
And this is where modern neuroscience provides some of the most sobering insights into the chronicity of SUD. The pathogen alters the fundamental molecular architecture of the brain. Let's begin with intracellular signaling cascades.
Specifically, the CAMP-PKA-CREB pathway. Let's unpack the biochemistry of that, because this pathway really is the engine of neuroadaptation.
Okay, so when dopamine binds to a D1 receptor on a medium spiny neuron in the striatum, it activates a G protein. This G protein stimulates adenylyl cyclase.
Right.
Adenylyl cyclase then converts ATP into cyclic AMP, or CAMP.
And then CAMP does what?
This CAMP then binds to the regulatory subunits of protein kinase A, or PKA, freeing its catalytic subunits.
And just to clarify, a kinase is simply an enzyme that modifies other proteins by adding a phosphate group to them.
Precisely. The activated PKA translocates into the cell nucleus and phosphorylates a critical transcription factor called CRE, that's CAMP response element binding protein.
Okay.
When CRE is phosphorylated, it binds to specific DNA sequences and alters the transcription of target genes.
It turns genes on or off.
Yes. Now, acute occasional drug use causes a brief transient activation of CM. But chronic, repeated drug use massively upregulates this entire CAMP-PKA-CREB pathway.
It becomes completely hyperactive.
Right. And here is where we see the opponent process theory manifest at the genetic level. CREB acts as a molecular engine for tolerance and negative affect.
Wait, how does a transcription factor cause negative affect?
Because one of the primary genes that CREB upregulates is the gene that codes for dynorphin.
The dysphoria peptide we talked about earlier.
Exactly. Chronic substance use hyperactivates CREB, which pumps out massive quantities of dynorphin, which then binds to kappa opioid receptors, shutting down dopamine release and generating the crushing dysphoria of withdrawal.
So CREB is essentially the genetic brake pedal, but it's pressed so hard it breaks the system.
Perfectly stated.
But CREB activation eventually fades after a few weeks of abstinence, right? Yet craving persists for years. The chapter mentions another transcription factor that acts as a much longer-lasting molecular switch, delta FOSB.
Delta FOSB is truly fascinating because of its extraordinary stability. Unlike most transcription factors that degrade in hours, delta FOSB lacks standard de Grande domains.
So it sticks around.
It has a half-life of weeks or even months. With every subsequent dose of a drug, more delta FOSB accumulates in the nucleus accumbens.
Oh, wow.
Long after the drug and the withdrawal symptoms are gone, delta FOSB remains in the nucleus, continuing to alter the expression of proteins that physically reshape the synapse. It maintains the brain in a state of heightened sensitization to drug cues.
You mentioned reshaping the synapse, which brings us to structural plasticity. And this is where I really stumble when reading the chapter, because there's a glaring counterintuitive paradox here.
That spine density paradox.
Yes, the textbook clearly states that psychostimulants like amphetamines and cocaine increase the density of dendritic spines on medium spiny neurons. They make the dendritic trees more complex, more branched. However, opioids like heroin and morphine decrease spine density. They aggressively prune the branches. I'm just stuck on this paradox. If cocaine grows spines, but heroin shrinks them, how are they both resulting in the exact same clinical manifestation of a severe substance use disorder? I mean, how does the brain care so little about the actual structural direction?
It's a brilliant question, and honestly, it was a confounding paradox in the field for years. We assumed that more spines meant more addiction, or fewer spines meant more addiction.
So what's the resolution?
The resolution requires a paradigm shift. The vulnerability does not lie in the absolute number of dendritic spines. The vulnerability lies in the hyperreactivity of the actin cytoskeleton within those spines.
The internal scaffolding of the cell.
Yes. Dendritic spines are not static, rigid structures. They are highly dynamic, constantly changing shape, driven by a protein network called actin. Both opioids and psychostimulants, despite having completely opposite effects on overall spine density, they fundamentally increase what we call actin cycling.
Actin cycling.
Yeah, they alter proteins like profilin and cofilin, which regulate how actin filaments assemble and disassemble.
So the scaffolding is totally destabilized. It's like concrete that is never allowed to dry.
That is the perfect way to visualize it. The synapses are locked in a state of chaotic malleability. They are structurally unstable, so whether the patient has more branches or fewer branches is totally irrelevant.
What matters is the remaining synapses.
Yes, what matters is that the remaining synapses are dangerously primed to morph in response to a drug-related glutamate signal.
So when they encounter a cue,
When a patient encounters a drug cue, enzymes called matrix metalloproteinases, or MMPs, are rapidly released. These MMPs cleave the extracellular matrix surrounding the synapse, allowing the dendritic spine head to rapidly expand in a matter of minutes.
minutes.
Yes, massively potentiating the synaptic strength and driving an overpowering surge of craving.
The brain is physically expanding its connection to the cue in real time. That is terrifying. And if structural chaos wasn't enough, the chapter delves into epigenetics, the ghosts in the machine. We aren't just talking about changing the shape of the cell, we are talking about changing how the DNA itself is packaged.
Right. Epigenetics involves environmental factors altering gene expression without changing the underlying DNA alphabet. A primary mechanism here is histone acetylation.
Okay, let's break that down.
So DNA is wrapped tightly around proteins called histones. When enzymes called histone acetyltransferases add an acetyl group to the histone, it relaxes the chromatin structure, opening the DNA up so transcription factors can read it.
And drugs impact this.
Heavily. Drugs of abuse cause widespread changes in histone acetylation, effectively leaving certain addiction-related genes permanently unspooled and active.
But the most compelling epigenetic story to me was the role of microRNAs, specifically microRNA 212.
MicroRNAs are absolutely fascinating regulatory molecules. They don't code for proteins themselves. Instead, they bind to messenger RNA and either destroy it or block it from being translated into a protein.
So they're like the editors of the genetic code.
Exactly. Studies demonstrate that with chronic escalating cocaine use, the expression of miR212 in the striatum drastically increases.
And what is its target?
Counterintuitively, miR212 appears to act as a protective homeostatic shield.
Really?
Yes. The escalation of miR212 activates more CREB, but in this highly specific feedback loop, it ultimately suppresses the production of BDNF, brain-derived neurotrophic factor.
Wait, BDNF is usually the miracle grower of the brain. In depression, we want more BDNF to encourage neuroplasticity and healing. Why is suppressing it protective here?
Because in the specific context of the VT8 and ACT reward circuitry, BDNF acts as a pro-addictive modulator.
Oh, I see.
It facilitates the aberrant learning and the physical entrenchment of the compulsive habit. By aggressively suppressing BDNF translation, miR212 is the brain's desperate attempt to put up a firewall against the escalating loss of executive control.
It's a microscopic war happening in the striatum, the genome fighting to protect itself from the chemical pathogen.
It really is.
So we have a brain where the physical scaffolding is chaotic, the epigenetic switches are stuck in the on position, and the dopamine baseline is in the absolute basement. When that patient leaves the detox facility and walks back into their old neighborhood, how do those microscopic changes trigger a macroscopic behavioral collapse? I mean, how does a relapse actually happen?
Relapse is the defining clinical hallmark of the chronicity of SUD. In translational neuroscience, we model this using reinstatement paradigm.
Reinstatement.
Yeah, an animal is trained to self-administer a substance by pressing a lever. Once the addiction is established, we implement extinction training. The animal presses the lever, but no drug is delivered.
So eventually the behavior extinguishes. The animal is abstinent.
Exactly. But the molecular memory of the drug is still hardwired. We then test what stimuli will cause the animal to reinstate the behavior, to start frantically pressing the lever again. And we identify 3 primary triggers. What are they? A drug prime, a conditioned cue, or a stressor.
Okay. The drug prime is the most straightforward. The patient has a momentary lapse, takes a tiny taste of the substance, and it immediately reactivates the circuitry.
Yes, a small priming dose causes a massive release of glutamate from the prefrontal cortex down into the core of the nucleus accumbens, violently waking up the dormant habit circuitry.
But cue-induced relapse introduces a phenomenon that really defies all intuitive logic, and it's vital for clinicians to grasp. It's called the incubation of craving.
Right.
Because if a patient stops using, you would logically assume that their craving is highest on day 1, and then it slowly dissipates over time. But incubation means the craving actually waxes. It grows stronger and more intense over weeks and months of abstinence.
It's a cruel biological trap,
Right.
and it completely reframes why patients so frequently relapse just when their families and clinicians think they're finally safe. The mechanism behind the incubation of craving is tied directly to those hyperplastic synapses we discussed earlier.
The actin cycling ones.
Exactly. During the early days of withdrawal, the brain generates what we call silent synapses in the nucleus accumbens.
Silent synapses. How can a synapse be silent?
Well, these are synaptic connections that possess NMDA glutamate receptors,
Yeah.
but they completely lack NMPA glutamate receptors.
Okay.
Under normal resting membrane potentials, NMDA receptors are blocked by a magnesium ion. So glutamate can be released, but the postsynaptic neuron just doesn't depolarize. The connection is structurally present, but functionally completely silent.
I picture this like a dormant volcano. On the surface, during the first week of detox, everything looks quiet. The patient seems stable.
But underneath, the pressure is building. Because over weeks and months of protracted abstinence, those silent synapses undergo a dramatic transformation. They are suddenly populated with a highly specific, dangerous type of receptor,
Which is?
calcium-permeable AMPA receptors, or CT-AMPAs. Normal AMPA receptors contain a GluA2 subunit that blocks calcium. But these new receptors lack that subunit. They are highly conductive and highly permeable to calcium.
The magma pooling in the chamber.
Exactly. The synapses have undergone massive silent potentiation. So 60 or 90 days into sobriety, the patient is feeling confident. They walk past a street corner where they used to buy drugs, or they see a piece of paraphernalia. The amygdala processes that cue and fires a glutamate signal into the nucleus accumbens.
And because the synapses are now loaded with these hyperconductive CPA-MPA receptors,
The calcium influx is catastrophic. The system simply erupts. The cue reactivity and the subjective experience of craving are exponentially higher than they were on day 3 of detox.
when you explain it like that the brain is secretly building a trap of calcium-permeable receptors 60 days into sobriety, it just breaks my heart. It removes the moral failure from the relapse entirely. It is a biological ambush.
It truly is.
And then beyond cues, there is stress-induced relapse.
Stress is arguably the most insidious and pervasive trigger of all. And biologically, a severe stressor acts on the brain in almost the exact same way as a drug cue. An acute stressor activates the HPA axis, but more importantly, it deeply activates that extended amygdala network we mapped earlier.
So it floods the circuitry with corticotropin-releasing factor and norepinephrine.
Yes. And because the patient's dopamine system is already hypofunctional from the prolonged withdrawal, that massive stress signal cuts through the noise perfectly. It drives them back to the substance not for pleasure, but via negative reinforcement. They need to shut off the excruciating stress response.
That is the vicious cycle, isn't it? Stress triggers the extended amygdala, driving craving, driving use, which further depletes dopamine, making them even more vulnerable to the very next stressor. Now, we have to add a critical contextual layer here, because this exquisite neurobiology doesn't exist in a vacuum. It interacts intimately with the patient's endocrine environment. The chapter makes it abundantly clear that the trajectory of a substance use disorder looks fundamentally different based on biological sex.
This is a crucial area of addiction medicine that historically suffered from a severe male-centric bias in both research and clinical application.
Right.
The epidemiological data is quite clear. Men have historically exhibited higher absolute prevalence rates of SUD, often cited at a 2-to-1 ratio. However, female patients exhibit a phenomenon known as telescoping.
Telescoping, meaning the clinical timeline from first exposure to severe pathology is compressed?
Drastically compressed. Women tend to transition from initial casual use to a severe treatment-resistant substance use disorder much faster than men do. Furthermore, female patients typically present with greater medical and psychiatric severity, more intense withdrawal syndromes, and significantly higher cue-induced craving upon relapse.
And we can trace this directly to the activational effects of gonadal hormones, right? The biology of estrogen and progesterone.
Exactly. Estrogen, which peaks during the follicular phase of the menstrual cycle, is a highly potent neuromodulator. It interacts directly with the methylimbic dopamine system.
How so?
Estrogen heavily potentiates the euphoric and subjective rewarding effects of psychostimulants. It enhances dopamine release and alters dopamine transporter function. In animal models of addiction, high estrogen states actually facilitate the rapid acquisition of drug self-administration at lower doses.
So estrogen acts as a biological amplifier for the drug's reward signal. It makes the high hit harder and faster.
It does. Conversely, progesterone, which dominates the luteal phase of the cycle, tends to exert inhibitory effects. It blunts the dopaminergic response and reduces the subjective reward of the substance.
That's a profound fluctuation.
And furthermore, neuroimaging shows that women, on average, possess a higher baseline capacity for striatal dopamine synthesis and reuptake compared to men. their basal dopamine tone naturally fluctuates with their hormonal cycle.
Which creates distinct shifting windows of profound neurobiological vulnerability to both initial drug reinforcement and subsequent cue-induced relapse.
Spot on.
I want to pause on this because the clinical implication is massive. If a standard clinical approach treats a male and a female patient identically, utilizing the exact same timeline, the exact same relapse prevention strategies, without ever considering these hormonal phases, that approach actively fails female biology. I mean, a female patient's cycle might dictate that her cue reactivity and craving intensity are astronomically higher in week 2 of the month compared to week 4.
It's the absolute definition of precision medicine and psychiatry. You cannot treat a neurobiological disorder without understanding the endocrine milieu in which those neurons reside. Assessing risk and tailoring interventions absolutely must account for these hormonal fluctuations.
Right. Let's transition fully into the consultation room then. How do we take all of these dense neurobiological models, the hypofunctioning dopamine receptors, the hyperactive extended amygdala, the hyperplastic actin cycling, and apply them to the human being sitting across from us in distress? Let's re-examine standard treatments through this mechanistic lens.
Okay. When we prescribe medications like buprenorphine or methadone for an opioid use disorder, we are heavily fighting against a massive, stigmatizing public misconception that we are merely replacing one drug with another.
I hear that all the time.
And it is scientifically illiterate. What we are actually doing is biologically stabilizing the severe allostatic state.
We are addressing the hypercataphia.
Exactly. Take buprenorphine. It's a partial agonist with an extremely high affinity for the mu-opioid receptor, but a low intrinsic activity.
So it binds tightly, blocking other opioids from attaching, but it only partially activates the receptor.
Right. This provides just enough tone to satisfy the receptor, shutting down the massive CRF and dynorphin storm in the extended amygdala without producing a euphoric high. It pulls them out of the allostatic pit so their prefrontal cortex can actually come back online and function.
You can't do therapy with a patient whose amygdala is screaming. And speaking of therapy, how does cognitive behavioral therapy, or CBT, map onto the biology?
CBT is effectively physical therapy for the frontotemporal cortical circuitry. Through repeated cognitive exercises, we are attempting to strengthen top-down executive function. We're trying to rebuild the prefrontal cortex's ability to exert inhibitory control over those hyperactive limbic drives.
And therapies like contingency management, where patients receive tangible vouchers or prizes for negative drug screens.
That explicitly targets the compromised mesolimbic dopamine circuitry. We are providing a competing, powerful, natural reward to slowly rebuild normal dopamine tone and retrain the approach circuitry.
Let's apply this clinical reasoning to 2 specific vignettes based on the text. First vignette. A 23-year-old woman presents to the clinic. She has a history of intravenous heroin use and a deeply significant history of sexual trauma. She is currently in early, acute withdrawal.
The clinical reasoning here must be hierarchical and neurobiologically found. Naturally, establishing environmental and psychological safety is paramount. But from a pharmacological perspective, she is in acute early withdrawal. Her extended amygdala is wildly overactive. Her hypercataphia is peaking.
Right.
Agonist stabilization, utilizing buprenorphine or methadone, is urgently required to re-equilibrate the system.
Let me push back, because a layperson might ask, if the ultimate goal is to get her off illicit opioids, why is naltrexone, which is a full opioid antagonist, an opioid blocker? Why is that the wrong move right now? Why use a partial agonist?
Because of the precise mechanisms we've spent the hour exploring. Her brain is currently in a state of catastrophic allostatic debt. If you administer a full antagonist like naltrexone to her mu-opioid receptors in the first 7 to 14 days of abstinence, you will violently displace any residual endogenous or exogenous opioid tone.
So you will plunge her extended amygdala into severe overdrive.
Yes, you will precipitate explosive, agonizing withdrawal. The stress peptides will flood her system, her distress will peak, and a relapse is virtually guaranteed. We must jointly re-equilibrate the tone first with an agonist.
And we cannot ignore the trauma either.
Absolutely not. Her history of sexual trauma means her HPA axis and her extended amygdala are likely already basally hyperactive due to untreated PTSD. The trauma acts as a permanent stress-induced relapse trigger. If you do not address the trauma through targeted psychotherapy, the addiction circuitry is constantly being primed for relapse by an endogenous cortisol and CRF storm.
Second vignette. A 55-year-old man, a highly successful business owner, presenting with escalating intranasal cocaine use. He reports profound, debilitating grief following the sudden loss of his partner 6 months ago. He is crashing.
With severe psychostimulant withdrawal, the primary neurobiological feature is not the violent physical sickness of opioid withdrawal, but rather a profound hypodopaminergic state.
Because his dopamine receptors are massively downregulated from the cocaine binges.
Right. Clinically, this presents as severe anhedonia, psychomotor retardation, profound depression, and a critically high immediate risk of suicide. The brain literally lacks the neurochemical machinery to process natural joy or hope.
So how do you treat that biologically? I mean, we don't have a methadone equivalent for cocaine.
We lack a perfect replacement therapy, yes. However, the treatment requires gentle dopaminergic support to mitigate the crash. Often, clinicians will utilize a medication like bupropion off-label.
Bupropion is a norepinephrine dopamine reuptake inhibitor.
Yes. It weakly blocks the reuptake of dopamine, gently elevating the baseline tone in the synapse. It provides a soft landing. But simultaneously, he requires culturally sensitive, intensive grief counseling. The loss of his partner is a massive, crushing stressor that is driving his negative reinforcement loop. He's using cocaine to artificially escape the emotional agony of grief.
It is entirely about mapping the clinical symptom to the underlying circuit. Okay, before we move to our review questions, I want to leave you with a final provocative thought. We've talked extensively about epigenetic changes, histone acetylation, microRNAs altering BDNF, and how they physically change the brain within a single patient's lifespan. But the literature is currently probing a chilling frontier: transgenerational epigenetic inheritance.
It is arguably one of the most controversial yet fiercely fascinating areas of modern molecular biology.
We have to ask, could a parent's severe chronic substance use disorder physically alter the epigenetic packaging within their gametes? I mean, are they passing a primed neurobiological vulnerability, a pre-hijacked survival compass, to their children before they are even conceived? It completely reframes our understanding of generational trauma and the cycle of addiction in families.
It really does.
It implies the biological echoes of this disorder could span generations. Something for you to ponder long after this deep dive is over.
It is a sobering reminder that we are treating an entity with profound temporal reach.
All right, let's reinforce the core concepts we've synthesized today with a rapid-fire clinical reasoning block. I'll ask a single best answer style question, pause for a moment so you can lock in your answer, and then our expert will deliver the precise answer and the biological rationale. Ready?
Ready.
Question 1, focusing on mechanism. Which substance indirectly increases dopaminergic transmission in the ventral tegmental area, primarily by inhibiting local GABAergic interneurons? Take a second to think.
All right, got your answer. The answer is opioids. Opioids bind to G-coupled mu-opioid receptors on VTA GABAergic interneurons, opening potassium channels, hyperpolarizing the cell, and stopping the release of GABA. This removes the inhibitory clamp, allowing dopamine neurons to fire rapidly.
Question 2, conceptual mapping. The transition from voluntary goal-directed drug use to automatic compulsive habit is mapped neuroanatomically to a shift in control between which two structures? Have a think.
The answer is a shift from the ventral striatum, specifically the nucleus accumbens, to the dorsal striatum. This transition is mediated by ascending, spiraling feed-forward loops connecting the midbrain striatum and prefrontal cortex, which entrench the motor habit independently of the reward value.
Question 3, mechanism of withdrawal. During withdrawal phase, which specific neuropeptide acts on kappa opioid receptors within the extended amygdala to decrease dopamine transmission and produce profound dysphoria? Take a moment.
That would be dynorphin. It is the primary stress peptide, alongside CRF, released in the extended amygdala that enforces the miserable, hyperketific state of the anti-reward system.
Question 4, conceptual chronicity. The phenomenon known as the incubation of craving over protracted periods of abstinence is heavily associated with the delayed insertion and increased expression of which specific receptor type in the nucleus accumbens? Pause to consider.
The answer is calcium-permeable AMPA receptors, or CPMPAs. Over weeks of abstinence, previously silent synapses are populated with these highly conductive receptors lacking the GLUA2 subunit, rendering the brain explosively, dangerously reactive to environmental drug cues.
Question 5, theoretical models. The hedonic allostasis theory posits that in severe chronic substance use disorder, the behavior is maintained primarily by which form of reinforcement? Have a think on this one.
Negative reinforcement. The patient is no longer seeking the hedonic high they are compulsively using to avoid or alleviate hyperkataphia, the persistent biologically enforced state of extreme emotional distress and low dopamine tone.
Question 6, clinical reasoning. In a patient undergoing acute opioid withdrawal, for example, presenting on day 3 of abstinence, why is the initiation of naltrexone absolutely contraindicated? Take a final second.
Because naltrexone is a full opioid antagonist. Initiating it during acute withdrawal will displace any residual opioid tone at the mu receptors, precipitating severe, catastrophic withdrawal. It drastically worsens the patient's allostatic state, floods the extended amygdala with stress peptides, and drives the patient towards immediate relapse.
Brilliant. Thank you for dissecting this incredibly complex landscape with us. Here are the details of this episode.
Neuroscience of substance use disorders. Byline. Hijacked reward when survival circuits are redirected towards compulsion.